Welcome everyone. Today we are going to learn about CFTR protein. The full form of this protein is cystic fibrosis transmembrane conductance regulator. Now the mutation of this protein can cause cystic fibrosis. Now this ambiguous term indicates that during the discovery of this protein, scientists weren't sure about the precise function of the protein. But in 1989, when the gene that is responsible for cystic fibrosis was isolated and sequenced, scientists observed that this protein is actually falls under ABC pump. ABC means ATP binding cassette proteins. So this protein, and further research proves that this proteins actually looks like ABC pump but works like a channel. So it actually like a hybrid protein. So this CFTR protein transport chlorine ions, also bicarbonate ions. Now, in my previous video, ABC transporter or ABC pump, I have mentioned that all ABC transporters actually contain two transmembrane domain and two ATP binding domains. We call T domains and A domains. So because the CFTR proteins looks like an ABC pump, so it also contains two T domains and two A domains, but additionally it contains one regulatory domain. As you can see, this is an example of an ABC pump in eukaryotic cells. It is called multi-drug resistance proteins because this ABC transporters is present in the tumor cells that actually exhibit multi-drug resistance to the chemotherapeutic drug. So here two transmembrane domain and two ATP binding domain is present. But if you see the structure of CFTR, you will find there is an extra regulatory domain is present apart from these two T domains and two A domains. Now, what is the significance of this regulatory domain? Phosphorylation of R domain and ATP binding opens the channel. So as you can see, this is the closed conformations and this is the open conformations. When ATP binds with this A1 and A2 ATP binding domains, the channels actually opens, and the hydrolysis of ATP causes the conformation of a closed conformation of CFTR channels. Okay. So this protein CFTR expressed in apical plasma membrane of epithelial cells in the lungs, intestine, reproductive tract, also in the sweat glands and pancreas. Although cystic fibrosis affects various organs, the respiratory tract usually exhibit most severe effects, and you have to remember this point because I will explain the reasons behind these pathophysiological conditions, why the patients of cystic fibrosis shows the most severe effects of respiratory tract. Now what happens? Victims of cystic fibrosis produce a thickened and sticky mucus that is very hard to propel out of the airways, and patients actually suffer from chronic infections and inflammations which progressively destroy the pulmonary functions. Now let's see the reasons behind this pathophysiological condition of cystic fibrosis. I have drawn an epithelial cells. These cells actually line the airways of lungs, and these cells are called polarized cells because they have different domains in the plasma membrane. The upper portion is called apical plasma membrane that face the lumen of the airways, and the lower portion is called basolateral plasma membrane. The tissue is actually exchangeable with blood, so there is two different domains present. Also, there are different transporters present in the basolateral and apical portions of these cells. Now let me give the name. This is the one water channel present in the basolateral portions. This is sodium potassium pump present. This is the potassium channel. This is sodium, potassium and chlorine symporter. So as you can see, there is different transporter proteins present in the basolateral membrane. Also in the apical membrane of the cells, CFTR protein is actually present in the apical portions. There is sodium actually channels present, and there is water channels also present in the apical portions. Now what happens? I will start this mechanism by first sodium and potassium pump. Basically through the sodium potassium pump, 3 sodium actually export occur that comes out of the cells into these basolateral positions. So 3 sodiums comes out of the cells, and 2 potassium actually get inside of the cells, so import occur. Now the 2 potassium that come inside of these cells actually go out, that means export occur through these potassium channels, and that's why it actually creates a circuit. Now the sodium that actually actually comes out of the cells here from one sodium. The sodium potassium chlorine symporter actually works by the chemical concentration gradient generated by the sodium potassium pump. As you can see, this pump actually electrogenic. 3 sodium comes out and 2 potassium comes in. That's why in the vasodilatory position there is more positive charge is actually present, because more sodium is present outside, okay, and there is negative charge in this side. Now from these 3 sodiums, 1 sodium actually come inside the cells through this importer. So 1 sodium actually come inside these cells, and through the sodium import, as because they are symporters, so potassium also import occur. So 1 potassium come inside, and that potassium also come outside through the potassium channel. So there is a 2 potassium circuits present here. Now chlorine actually moves inside the cells through this importer. So I'm writing here 2 chlorine actually come inside, because through these CFTR channels this chlorine actually come out of the cell in the apical portions. That's why this outside of the cells actually becomes negatively charged, because more chlorine ions are present outside of these apical portions in the lumen. So this importer works due to this pump, because this pump actually generates the sodium concentration gradient. That means outside is higher concentration of sodium, that's why the sodium imports occur. And as because this is importer, so they actually, this sodium import actually helps the import of potassium and chlorine. Potassium come out of this channel, as because the more chlorine ions actually accumulate in the apical portions, sodium ions that is present higher in this portions, vasodilatory portions, sodium ions do not move through the cells, but it moves through the leaky tight junctions. So from these leaky tight junctions, sodium actually comes in these portions, because there is more negative charge present, so sodiums actually get attracted toward the apical portion. So basically here sodium actually starts to accumulate, okay, starts to accumulate. Now the presence of chlorine ions and sodium ions increase the osmotic force, so that water can move through this water channel and directly can come out into the apical portion. So what happens here, basically by this working of sodium potassium pump, potassium channel, this importer, water channel and CFTR and water channel in the apical portions, basically a fluid secretion occur. So what happens here, fluid secretion occur, fluid secretion, that means water secretion occur, but there is a balance of the secretion and reabsorption also occur. Not only water actually comes out of the cell, but reabsorption also occurs. So one thing you remember, fluid secretion and fluid reabsorption occur. Fluid reabsorption occurs. How fluid reabsorption occurs? Because there is a sodium channel, so sodium actually can come inside of the cell. Not only that, I have mentioned in my previous video that channels can actually transport the ions in both directions. So as chlorine can move out, when the concentration of chlorine actually becomes very high outside, then chlorine can also move inside of the cells. Okay, sodium moves inside the cells when sodium concentration increases outside. So by this way, the osmotic force in this apical portion is actually lowers. So at first it actually increases and then fluid secretion occurs, and after sometimes it actually decreases because the chlorine and sodium actually moves inside the cells. Then the osmotic force decreases, and by the way the water actually come inside through the water channel also. So by this way it actually helps in the reabsorptions. Now there is a balance of these secretions and reabsorption in the normal individual. So a thin layer of, so what happens here, a thin layer of fluid actually present in the upper portion of these epithelial cells. So a thin fluid layer is present in normal individual, and this fluid actually helps the cilia to clear the mucus and bacteria in from the airways. Now, what will happen if there is any problem in the CFTR protein? Then basically, if you see, the less amount of chlorine will come outside of the cell, so the chlorine actually becomes lower in concentration in this site, so potassium will not move into these apical portions, and as because there is low amount of osmotic force generated due to the problem in CFTR, then water will not actually move in these portions, apical portions, so the lung become too dry. So as I have written here, pathological symptoms that most severe effects on respiratory tract occurs. What happens, the cystic fibrosis patients produce thickened and sticky mucus, as because there is low amount of secretions of fluid, so it becomes thickened and sticky, and cilia is not able to clear the sticky mucus, so it is not easy to propel out the mucus, and that's why more bacteria grows and it causes the chronic infection which progressively destroy the pulmonary function. So CFTR protein mutations, if there is any mutations occurred, or if CFTR protein is not present in the plasma membrane, then this balance, remember the term I have mentioned here, there is a very intricate balance between this fluid secretion and reabsorptions. Now there are thousands of different mutations can be found in the CFTR protein that can cause the cystic fibrosis, but one of the mutation is very common to all the patients. About 1/3 of the patients, approximately 67% of all cystic fibrosis cases, has a single mutation in CFTR protein, that is deletion of one amino acid called phenylalanine at the position of 508. So this is very common mutations that are found, and this mutation actually occurs in the A1 binding of ATP binding domain, A1 ATP binding domain. So the mutant delta means here this amino acid is not present, so deleted 508 is temperature sensitive. Now this protein not folding properly at 37 degrees centigrade in the endoplasmic reticulum, so these proteins do not fold and remains in the endoplasmic reticulum where it is actually degraded, so it will not go to the cell surface or the plasma membrane for its proper functions. But scientists have found if the cells expressing this mutant CFTR are incubated at 25 degrees centigrade, that means lower temperature, the protein folds and accumulates normally on the plasma membrane, and you will be surprised to know that this mutant protein can actually work as well as the wild type CFTR can function. So there is no problem in the transportation of the ions, but basically the deletion of this phenylalanine at the 508 positions cause them to not fold properly in the endoplasmic reticulum. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching and always keep learning.